Is germline gene therapy a future for cystic fibrosis? Is germline gene therapy a future for cystic fibrosis? This is an intriguing question that touches on the cutting edge of genetic research. Germline gene therapy involves making changes to the genes in sperm, eggs, or early embryos. This means any corrections made could be passed down to future generations. However, as of now, this approach is not practical or approved for treating cystic fibrosis. The current focus is on somatic gene therapy, which targets the affected cells in a patient's body without altering their germline. Cystic fibrosis is caused by mutations in the cystic fibrosis transmembrane conductance regulator gene. These mutations lead to issues with chloride ion transport, resulting in thick mucus buildup in the lungs and digestive system. Current treatments include cystic fibrosis transmembrane conductance regulator modulator drugs that help improve the function of the faulty protein for many patients. However, about 10 to 15% of individuals with cystic fibrosis do not benefit from these treatments, highlighting the need for alternative therapies. Recent advancements in gene therapy for cystic fibrosis are promising. Researchers are exploring ways to deliver a functional copy of the cystic fibrosis transmembrane conductance regulator gene directly to airway cells. One example is a first-in-class inhaled lentiviral gene therapy, known as by 3,720,931, which is currently in clinical trials. This therapy aims to improve lung function and reduce disease progression in patients who do not respond to existing cystic fibrosis transmembrane conductance regulator modulators. The therapy delivers the cystic fibrosis transmembrane conductance regulator gene to respiratory epithelial cells through inhalation. Early studies suggest that expressing the cystic fibrosis transmembrane conductance regulator gene in just 9 to 15% of airway cells may provide clinical benefits. Importantly, this approach does not alter the germline, which is a significant consideration. Germline gene therapy for cystic fibrosis faces numerous challenges, including scientific, ethical, and regulatory concerns. Editing the germline raises questions about unintended consequences and the potential for heritable off-target effects. Therefore, current research prioritizes safe and effective somatic gene therapies that can be administered to living patients without affecting their offspring. In addition to viral vector therapies, researchers are also investigating gene editing platforms, such as CRISPR-based approaches, which aim to correct cystic fibrosis mutations directly in lung cells. These methods are still in the early stages of development but hold promise for providing lasting benefits with one-time treatments. In summary, while germline gene therapy presents a theoretical future possibility, the current focus for cystic fibrosis is on somatic cell, gene addition, and gene editing strategies. These approaches aim to improve or correct cystic fibrosis transmembrane conductance regulator function in patients without altering the germline. As research continues to evolve, it is essential for individuals to consult healthcare professionals for personalized medical advice and to stay informed about new developments in cystic fibrosis treatments.